Welcome back. I have to leave my phone on because there might be a delivery coming that I have to talk to them about. So that'll be fun if that happens during our class. So here we are. What day is it? It's a Tuesday. So what are we going to do this week? Today, we'll see that we'll finish talking about Chapter 18. We have the stem cell assignment you're working on. That's a big part of Chapter 18, and that's due on Thursday. And then what we're going to be talking about is continue to talk about programmed cell death, and especially apoptosis as our favorite example of programmed cell death. And so we're going to move on, and it's the final chapter in our book, and I think it's just called Cancer. So this last section, our book is divided up into 4 sections, and the last section is these 4 chapters where we jump to the cell signaling, then we have cell cycle, programmed cell death, and cancer. And so, it's the last chapter in our book because our book is ramping up the whole way to this section and that final chapter. And so we got to read through that, and then work on your term paper. You're kind of doing that somewhat on your own, but bring questions to class or ask me them. And remember to work on the shared documents that I have. If you haven't tried to open them yet, you really should. Make sure you're set in understanding that thing. So then again, we have that thing, and then we have our second exam a week from this Thursday. And you'll see that we're still learning a bunch of new material, but we'll be fine because we've been foreshadowing these chapters and this part of our course since the very first day. So we got to keep rolling with that, and that's where we're at. And this end of this week is when these applications are due for this Maine Health Institute for Research internship that occurs during the next school year. And if you can't meet that deadline, just let me know, because I'm the person who's in charge of making the connection between USM students and these scientists here at Scarborough and Maine Health Institute for Research. And it's a paid internship. Students get, for being down there from September to May, about 15 hours a week, they get about almost $9,000 of a stipend. So it's a really big effort on USM's part to build and maintain this relationship, and students have just had fantastic success. If you're going to be around here next school year, you should talk to me about it because it's the best research internship program I've ever experienced, and I've experienced a lot. So I'm really impressed with their welcome. I was down there yesterday, and USM Marketing was down there making a video of one of our students working in the lab down there because they're just like, this is super cool, and we have access to it. So that's that. Let me know about that. And the topics and style of the science down there is very much like what we talked about in our course. So I wanted to loop back to this, or not really loop back, or loop to the side of this because it's covered in the stem cell video that several students have already sent me pics of the drawings from the video. And it's covered in here, and it's about these adult stem cells. And I say our favorite example in our course are our muscle satellite stem cells. They can be used as an example in a general sense of what stem cells do throughout our lifetime. And so we know a lot about it. There's going to be changing gene expression as the stem cells develop into the specific types of cells. So muscle satellite stem cells, that's covered. Then we don't need to learn about the liver, right? And we don't need to learn about the skin and where the equivalent adult stem cells are located in our skin, but you're familiar with that's constantly regenerating. And I say the same is true here. These are the cells in our bone marrow and the stem cells in our bone marrow that a lot of people are familiar with that regenerate and replenish our blood cells all the time. And we don't have to be experts on this, but I realize that something's going to come up today, and some things have come up in the past. So I just want to guide us through this, just so everybody has a good sort of working appreciation of what we're talking about. And so that first cell up at the top, that's a stem cell, an adult stem cell in our bone marrow, and it's in a little microenvironment. It has neighboring cells that are covered in the video for the homework assignment. And then it's just constantly dividing, replenishing, and renewing our blood cells every day. And so let's just look at this. So we have this concept of a stem cell niche. So this is showing in our bone marrow. So in our bone marrow, within that is where these stem cells are located. And there's dividing to make precursors our whole lives. We don't lose those stem cells because they divide asymmetrically, so we're constantly maintaining the number of stem cells as we generate these precursor cells. And so let's just look at the cells that are produced, okay? So the first one is red blood cells. Sorry, I kind of messed that up, but red blood cells. Those, you gotta have a bunch of those new every day. They don't last very long. In humans, the red blood cells, the mature red blood cells, they don't even have a nucleus. They threw their nucleus away so they could be little and slip through our tiniest capillaries. So they only last usually about like 4 months, and they just die, okay? Then platelets, well, they're not even cells. There are pinched off portions of megakaryocytes in the bone marrow. They don't last very long because they're not even cells. Then the rest of the cells, there are a bunch of different kinds, are just called white blood cells. And in general, they're involved in our immune system. So our immunity and our ability to fight off infections and also deal with things like a twisted knee that gets swollen. That's our immune system also responding and causing inflammation and so forth. So just so you know this, we make lots of these cells every day. So in our bone marrow, there's a very dangerous game going on because these are normal cells that through our lifetime divide really fast. And that's dangerous because what if something happens, it gets disrupted. And so we want to pay attention to that and be worried if there's a mutation in the normal growth signals that are pushing these cells through the G1 to S transition of the cell cycle. The light is turning green for these cells very often, okay? And so that's what's happening in our bone marrow. And some of these cells continue to divide even after they've come out of the bone marrow. So they're not finished and they haven't fully differentiated. And so lots of proliferation in our bone marrow and even some after they come out. So I skipped that one because I tucked those in somewhere. So within our white blood cell population, we're learning a little bit about T cells. So there's a specific category of white blood cells called T cells, and we're learning about them because we're learning about that CAR T cell therapy. And so those cells are cells, white blood cells, that are in our blood, and they can go out into our tissues to fight infections, and they can stimulate inflation. They do a lot of different things. So T cells, they're just called T cells, and within the T cell population, there's a bunch of different kinds of T cells. Those are the cells that we're talking about, and when Sam talks about CAR T cell therapy. So they're white blood cells, they come out of those, okay? Then we want to talk, and we've talked very briefly about B cells. So another category of white blood cells are called B cells, just B, the letter B. And their role is to make antibody proteins. So they are going to release these proteins. They're going to make them, release them, and they'll be in our blood. And antibody proteins can bind to viruses, and they can kind of tag the virus so that T cells can find them better and wipe them out. Another thing that B cells can do is they can just block a virus. So they can neutralize a virus just by binding to it and blocking it from its ability to infect a cell. And so they call that neutralizing. So they could neutralize a virus like that. So B cells. And these are cells that come from the bone marrow. Lots of proliferation of the bone marrow. And as they come out, they are immature B cells. And they're ready to divide more. What happens is our body makes lots of these B cells to be ready to fight any kind of infection. And most of those don't find an enemy target, and most B cells are going to die after just a couple of months. On the other hand, if a B cell finds a target, it's stimulated to fully divide, fully differentiate, mature, go through some more cell proliferation. So the B cells is this pathway that we want to remember. And we make 1 billion of these B cells every day. That's a lot more cells we make every day than our total number of new skin cells. So everybody knows, oh, you're making new skin cells every day of your life. And this little AI overview seems to have, it makes mistakes because AI is stupid. And it seems to be off by order of 10. I think we make about 50 million new skin cells every day. We make way more of just these B cells, just to make antibodies. So think about these cells. The precursor cells are dividing really fast, much faster than our skin cells are dividing, to replenish them. Constantly dividing like that, okay? So MYC is normally activated. So remember when we talked about our skin cells, we said under a scab, it's all about the PDGF activating the RAS and the MAP kinase, RAF-MEK, or ELK pathway, and activating MYC expression. So in these precursor cells, in the B cell line or lineage, it's MYC, but it uses a different pathway, different signals, signaling molecules, different receptors, and different pathway. That's fine. We don't need to learn the pathway. Just recognize that it's different, and it leads to activation of MYC. So remember, we learned that MYC our cells are trying really hard not to express it. They don't want to let the polymerase transcribe it. They don't want to make the protein. They don't want the protein to be stable. They don't want the RNA to be stable, right? And so we saw that there's a certain kind of cancer for our first exam called Burkitt's lymphoma. And in Burkitt's lymphoma, there's a translocation between the transcription unit of MYC and the antibody cis regulatory region. And there are really there's a few different kinds of antibody genes, so the translocation can be a little different. That's fine. But it's key concept. And just want to remind you of that what's going to happen is that MYC is normally stimulating the proliferation of the B cells, and it can get out of control by overexpressing MYC, okay? So that's something we know. And it's going to come up again today, coming down this lineage and being like, whoops, you're messing up, okay? So that's just the image we had for our first exam, is you can get cancer, Burkitt's lymphoma, by just overexpressing MYC along these B cells, okay? All right, so that's just a side note to remind you of that, kind of help set up something that's going to bounce out today, where we're going to get back to those B cells today and a whole different cancer story that's associated with programmed cell death. So we've been talking about programmed cell death, chapter 18, and we really want to focus on the molecular story. What does it mean when a cell goes through apoptosis? And there are some hallmark features of apoptosis, and our book lists them. So we're going to learn especially this idea of the flipping of the phosphatidylserine from the inner leaflet to the outer leaflet of the cell membrane in a cell that's going through apoptosis. And that's the signal for another cell to phagocytize, to eat the dying cell. And it gets activated by this scramblase protein. That scramblase protein flips the phosphatidylserine. Then we're talking a little bit about how the DNA is going to start getting chopped up. That's by the activation of an antibody DNase. And then we'll talk as we go along a little bit about a role for the mitochondria. Those are the most important things. These, the book talks about very briefly. Maybe there'd be a question on your exam, like a 2-point question or something that's in your, in the book, but not in your, not something we talked about in class. But probably not, because we have such nice examples, such memorable examples. And so we talked through these some. There's the DNA getting cut up. And we talked about that, how sensible that idea is, because what's being cut up into is sort of bite-sized pieces that are about the length of DNA that's protected by a nucleosome. And so the DNase is first cut between them, and they just cut, and eventually they're just going to cut it all up. Then you can see in this figure, they also show a picture of a micrograph, pictures under a microscope. And here's a normal cell, and so as the DNA is getting cut up, another thing that happens is the nucleus collapses into a bunch of little, tiny, really darkly stained little droplets. And so scientists have been able to see that for a long time, and they're like, that cell is dying in a weird way, okay? So that's happening, and these are parts of that pathway, okay? And what we started talking about a little bit is there's an enzyme. It's an enzyme, it's called caspase, and it's a family of enzymes, so they come with numbers, caspase 9 and so forth. We don't need to pay attention to the numbers. We know it's a gene family. And all of the caspases are enzymes, and they all have a similar enzymatic action. And the enzymatic action of caspases is their enzymatic activity is they are called proteases. And so what they do is they cut proteins. And there are lots of different proteases in our body. And some proteases are involved in our metabolism. And they just break the proteins we eat down to individual amino acids. Caspases are different. Caspases are a class of proteases that have a very specific target. And so, for example, when we look at the caspase activity on the scramblase, right now we have scramblase in all our cells. It's waiting. And the scramblase looks like this. It's a protein, and then it just has an extra bit on it. And that extra bit is inhibiting the activity of scramblase, and the caspase just cuts that piece off. So it's a protease, it cuts proteins, and it cuts target proteins, and it just cuts a piece off, and it releases it. So now we have that little bit shorter scramblase, and it's now activated. It lost that inhibitory piece. So the substrate for a caspase in this case is the inactive scramblase. It's always present. And the product of the enzymatic reaction is the scramblase that's now active and a piece of the protein that got cut off. And that's how caspases activate cell death, is all of our targets, in general, their targets have this inhibitory part of the protein, and the caspase needs to cut it. So they're enzymes that activate, and an enzyme can do a lot because it just does that same reaction over and over again. So a few caspase molecules can cut lots of scramblases and activate them. And so that's the case in general with these caspases. They're proteases. And when they're activated, the cell is now going through programmed cell death. And so there's a term, and the book uses it a little bit, but it's kind of a loose term, and these would be called effectors. And that just means they are the action protein in this story. That when these guys are active, the cell's going through cell death. If you were asked on the exam, name two effector proteins we talked about, you'd say caspases and CDKs. So CDKs are also big-time effectors. Remember, we emphasized CDK1, it'll phosphorylate its helper kinases, and Polo and Aurora, and then together, those effector kinases, they just phosphorylate things like histones. And when histones are phosphorylated, they fold up tighter, and you see the chromosomes. The chromosomes literally connect. That's action, real action item. So these are effectors. They're getting the job done. The reason that people would really kind of emphasize this is because of the fact that these are in our cells all the time. So they're ready to go. That's true for those CDKs also. Remember, we didn't talk about them getting increased. They're always there, right? So it's a tricky, kind of dangerous game, to be saying, I'm just ready to go into the cell cycle. Are you ready? Like that, yep. Or I'm just ready to die, And it's just a kind of a indication of just how poised the cell is to just take itself out. We're going to evolve this idea of having a multicellular organism. We better be ready to take out these individual cells. Like they're a living cell, but they need to be ready to just get wiped out. Okay, so these are effectors, and so what we must want to talk about a lot is we must want to talk about the regulation of these. So upstream regulation, like that. That's what we want to focus on today. And so one word you need to learn is caspase. And so in this figure, this is a nice figure, is that, caspases. And see that arrow is like, well, what's regulating those caspases? They're the effectors. When the cell activates those, the cell's going to die. And it's going to divide by these cellular mechanisms. like that, okay? So we're gonna work on that. And so here's this figure, and I don't, the figures, it doesn't look like, it doesn't really look that great, I don't think, as a figure showing what's going on. But we can work a little bit backwards. So there's a apoptotic cell. And here's the activation of the effector protein. Why don't they just call that a caspase? It's the caspase. Activation of caspase, okay? And so look upstream of this. Upstream of this is this regulation, and you'll see that we're gonna look at this regulation carefully, but very specifically. And there's lots of upstream regulation that occurs, but in our case, we wanna focus on one protein. And it's this protein here. No, it's not really that protein there. So we'll get to that protein, yep. Then further upstream of that is the idea that there's a cell signal. And remember, the cell signal could be internal. Oh my gosh, there's DNA damage. Or it could be external. Oh my gosh, the T cell is hugging me and I think I might be a cancer cell and should wipe myself off. So upstream signals are either extracellular signaling molecules or intracellular signals like DNA damage, like that, okay? Through this regulatory pathway, that's really complicated. And there's a key concept, and the key concept is simply this, that in the upstream activation, there are activators and inhibitors of cell death. And the general term for them is, if activators are called pro-apoptotic activators, that's a satisfying term for a student, isn't it? So we have some pro-apoptotic activators, and we have some anti-apoptotic inhibitors. Apoptotic inhibitors. And the one name that we want to be solid on is an anti-apoptotic inhibitor, and it's called BCL2. And it's an inhibitor of programmed cell death. It's an inhibitor of caspases. And it's always in our cells, and it's holding back. There are other anti-apoptotic inhibitors. This is one. This is a famous one, and it's BCL2 stands for B-cell lymphoma, because this protein was discovered when people were studying a certain type of cancer. And it was a lymphoma. Lymphomas are cancers of the blood. They don't make solid tumors, they're just the cells circulating in our blood. So lymphomas and leukemias and myelomas are all that type of cancer. There's cancers in the blood, they don't make a solid tumor, like a brain tumor that could be removed by surgery perhaps. So BCL2 is an anti-apoptotic regulator of caspases and apoptosis. All right, so here's what happens in this. BCL2 was the first one of these that was discovered, and then it was discovered that there's a family of genes that are paralogs with BCL2. And everyone calls the family the BCL2 family. Let me just find that and see. So this is shown in our book, and we need to learn the name of one of these proteins, and it's BCL2. So BCL2 gene family. They're gene family duplicates by thing. And here are their names, and they have goofy names, and you just don't have to worry about them. Even, there's not gonna be a 2-point question. They're like, hey, what is one of those other names? BCL2 is anti-apoptotic. These are pro-apoptotic. Same gene family, opposite action. So that upstream regulation is complicated just within this gene family. This gene family evolved to have some that are pro-apoptotic and anti-apoptotic. And you'd be like, how could that ever happen? And the way I describe it is, well, just imagine you have a gene family of screwdrivers, and some can only turn the screw in, and some can only turn the screw out. They're very, very similar. It's just a question of which way they turn. So that's what happened in this gene family. And so it's frustrating because it's like, whoa, that's weird. And it's frustrating because there's a bunch of them. So there's just a little blizzard of complex regulation upstream of the caspases and downstream of intracellular signals like DNA damage or extracellular signals like signaling molecules. There's this, and it's happening in the cytoplasm, and it's just complicated. What do we want to remember? BCL2 is a anti-apoptotic regulator, and it is the name of a large gene family that have members that are both pro and anti-apoptotic. If we just take a quick look, and again, I'll just point out Puma and Noxa, but don't remember those names, even though maybe Puma is cute and memorable. I'll just point this out. Let me just show you this. Here's that P53 hyperphosphorylation. So remember, P53 gets phosphorylated, and initially it binds to the cis-regulatory region of P21, activates expression of the P21 gene. The P21 gene is a coding gene. It codes for P21, which is a small inhibitory protein that works like a school patrol officer to stop the cell cycle at start. If the DNA damage is not corrected, P53 gets hyperphosphorylated and binds to new target sites like the cis-regulatory region of these BCL2 gene family members, Puma and Noxa, that are pro-apoptotic members. So that's the full story of P53. You don't have to remember Puma and Noxa, remember pro-apoptotic. Right? They're going to push the cell toward that apoptotic fate. There's this like mixture of those proteins. Some are anti-apoptotic, some are pro-apoptotic. It's just going to make more of the pro-apoptotic factors. And I skipped over some of these slides, so let's just go back to this and dig in on this some. So this is just something the book talks about. This is from a different textbook, and it shows, what it shows is these colored balls, and these colored balls are orthologs, and they're orthologs in every animal and basically every plant. And so it shows over here the scientists who first discovered these regulatory factors and the regulation of programmed cell death. They were studying a little tiny worm that's called a nematode that has become like a model organism, kind of like an equivalent of a fruit fly. Just a real simple little worm. It's almost too small to see, and it normally lives in the soil, and it doesn't really do anything particularly awesome. Yeah? And so they just use just a powerhouse genetic strategy down at MIT, in one lab at MIT, and the picture is in the book, a slide picture of the leader of that lab, and Bob Horvitz, and they identified these in a pathway. And this is caspase. So if you go over there to mammals, caspase. So they identified the effector, and then they identified these regulatory molecules, including this. They called it SED9. We never would. That's fine. We call it BCL2. And you can see that in this little worm, it's a relatively simple regulatory pathway. They're really tiny. They only have about 1,000 cells. They don't have to do a lot of programmed cell death for a lot of different reasons. We do. Notice, though, that some of these have arrowheads. They're activators, stimulators, and some are repressors. So you see that BCL2 in these simple worm. It's inhibiting an activator of caspase. Like that. So it's putting the brakes on. It's anti-apoptotic. And these guys have a simpler apoptotic regulatory pathway because they're a simpler organism. Here's fruit flies. Fruit flies, there are orthologs of these, BCL2 and caspase, the ones we really care about. And they don't use those names because they're goofy fruit fly geneticists. And so they use names like Reaper and Grim and stuff like that. This goofball. Sickle, you know, because it's all about death, right? And then in mammals. And so in mammals, see that, you know, we're going to have an upstream signal, and it's just complicated. There's a lot more factors than that in us, right? Like that. That's fine. It's fit. It makes sense in evolution that it'd be that way. Sam, do you have a question?
This slide says learn these names with
Yeah.
arrows pointing at CED9 and CED3.
Yeah, that's because I use the same slide in developmental biology where we talk a lot about nematodes as a model organism. Learn these names and point to the caspase 9 and the BCL2. Yep.
Is it always caspase 9?
No, caspases are in a family, and that's
Yeah.
just naming one of the family members.
But
Caspase 9 is a famous one.
Okay, are we specifically talking about 9?
Nope.
No.
We just started learning caspase and recognizing that they're in a gene family of caspases, that the genes are numbered. And that's nice for student learning. They're all caspases. Notice in the BCL2 gene family, they didn't do that. They didn't just number them. They named them things like Puma and Noxa. And you're like, come on, please. You know? Yes. Hi, Alex.
So you don't want us to like fully
Nope.
have this.
Just kind of see the visual. And the visual is colored orthologs in these animal models. And then the simpler kind of regulatory pathway in a simpler organism, you know, it's just got 1,000 cells, normally only lives 2 weeks, right? Are they too worried about programmed cell death? No, but look, they are. They have it. There's this programmed cell death even in really simple organisms. So really simple organisms in early multicellular evolution evolved this capacity to wipe out individual cells like that. It's not always because they're out of line. Sometimes they're perfectly normal cells, but they just evolved to play a role, and when they're done, get out. You're done. Yep. Yep. So I showed this slide just as a visual, and again, BCL2 anti-apoptotic and caspase effector. Yep.
I'm just struggling to understand how it can be an inhibitory that activates something. Like between like the CED9, you said that that's like inhibits the CED4?
Yeah. Yeah.
I just-
So caspase is the gun. This protein is the bullet. BCL2 is the safety.
Okay. So it's already there. It's just like it's like limiting basically its functioning.
Mhm.
Okay, that makes sense. Also, I don't know if now's a good time to ask this question, but should we be trying to remember the name of scientists? Like I noticed
No.
in the stem cell video, like
No,
you
except for
did-
me.
Okay. I actually have you cited in my paper, as funny as it is. It's like class with Dr. Champlin. Anyways,
Yeah.
but like you're talking
Yeah.
about one of the scientists a lot, and you're talking about his name, like in the stem cell video. So I
Mhm.
just didn't know if it was important to remember
Mm-hmm.
the scientist name.
Yeah, you should remember my name. And and you know, we're busy, but here's something that happened in my lifetime on this planet, and that is there's a Maine State Science Quiz Bowl for high school teams where they get together a team of like 4 students, and they're going to answer questions really fast about science, all aspects of science. It's really crazy because you know they buzz in and they're like, you know, you do some really complicated math equation and they just go, yeah, 2, it's 2, and then they're like, no, yeah, okay, yeah. So it's really interesting. So I like to judge that, and I read the questions, you know, and sometimes I make them up, and I'm like, and this one's from Star Trek, and all the people are all like, really though? You know, because that's not science, and I'm like, shut up. But anyway, so it's lunchtime, and they're eating, because they have it at USM, and they're eating at the cafeteria, and the teams will wear, they're high school kids, right? They'll wear matching shirts, you know, like that. And so here's a team, and they're sitting at their table with their backs to us, and they've got the name. It's like a jersey, like a sports jersey, and they got the last name of a famous scientist on their back, like Newton and Einstein and Maxwell, right? And one of the organizers of this statewide event is chatting with me, and he's looking at this one team that has those names on the back, and he says, what is going on there? And I look over there, and it's like Newton, Einstein, Maxwell, Planck, Champlin. I'm like, yeah. And I didn't explain to him, but it was my daughter, one of my daughters. Yeah, he was just blown away because he's like, how are you know, these are high school kids from all across the state, and how do you rank with Einstein? I was like, just take it easy, right? So we remember for the BCL2 gene family this idea of orthologs and paralogs, right? That's important for us to appreciate, you know, certainly for the final exam, maybe not so much for the second exam, but that's what's happening here. Those arose by gene duplication. It makes sense, right? The nematode has a relatively simple toolkit with just a standard screwdriver and a Phillips screwdriver, and the human has the screwdrivers too, but they've just diversified in more detail. And so it's a good example where it's like some are pro-apoptotic and some are anti-apoptotic. They're really similar, but they have just diversified their roles some. Yep. And so, yep.
Are these orthologs just in animals? Because
So I'm going to say that definitely caspases are in plants. Yep. And then I don't know so much about the details of the upstream regulation. But apoptosis is found in all plants and animals, and it's really significant. Yeah. Yep. Yep. And so I just included. Oh yeah. So there's that, and there our book is showing this, and it just I don't think it's just a very good visual to just be like I see we got these effectors and they're going to be regulated upstream. Okay. So there's that. We talked about and paralogs, right? Distinguishing between paralogs and orthologs, and how paralogs are gene family. Great. Okay. So now we're going to talk about this. So this is going to come up in a cancer story. That BCL2 gene is inhibiting programmed cell death. Could it be mutated and cause cancer? It doesn't seem like it could. We're used to oncogenes like MYC and RAS that are going to stimulate the cell cycle. So this is, I think, maybe the first gene that was identified in cancer where it was like, I don't know what I call this. I mean, it's kind of like a tumor suppressor or something. What's going on with the role for this gene and protein in this story? Okay, so we got 1 billion of these new B cells being made every day. Rolling, rolling, rolling. MYC is part of the story, keeps stimulating their cell cycle. And then this is true too. Remember, I mentioned this briefly. If those B cells don't get activated, they die after just a couple of months. So another story here is we make a lot and we kill a lot. And that's why we're making a lot, because a lot of them don't get activated. We don't run into that particular foreign molecule, so that cell doesn't get activated, so it gets cleared out. Who do you suppose is doing that? It's apoptosis, right? So we have lots of cells in our body that are going to live as long as we do. These do not. If the B cell gets activated, it doesn't die. That's how we have this immunity. When we get immunity to a virus that we get exposed to, like the cold or the flu or COVID, that's how our immunity is held, is the cells, B cells or T cells, live longer because they were activated. So there's an adjustment to their plan for apoptosis. They have a built-in timer that's just going to wipe them out unless they get activated, and then it's going to hold off, right? So that must be the regulatory story. So BCL2 is playing a particularly significant role because it's an inhibitor. And there are other inhibitors and activators, but in this story, it's a really important part of the inhibition of programmed cell death, okay? So BCL2. So now we're just going to focus on this and we'll talk a little bit about it, and it's going to roll into that there's a normal function for BCL2 here, and it's going to get disrupted by a mutation in lead to cancer, right? Okay, so B-cell2, B-cell lymphoma 2, coded by humans by the BCL2 gene, is the founding member of the BCL2 family of regulatory proteins that regulate cell death, programmed cell death, apoptosis, by either inducing pro-apoptotic or inhibiting anti-apoptotic apoptosis. Wikipedia is great. I actually donate, as a professor, I donate most of my salary to Wikipedia. True or false? False, right. I did send him $10 once, though, because my other daughter got listed on a Wikipedia page. And I was like, go, Wikipedia, go. Yeah, okay, so that's just good. It's good like college learning. BCL2 is specifically considered an important anti-apoptotic protein, but is not considered a proto-oncogene because it's not a growth signal transducer. Like that, right? BCL2 derives its name from BCL2 lymphoma, as it is the second member of a range of proteins initially described in chromosomal translocations involving chromosomes 14 and 18 in follicular lymphomas. So remember the idea that when you see under a microscope a cell in mitosis, you can see the chromosomes, and you can do a karyotype, and you can look at them. And scientists have been able to do that for 100 years. And for a very long time ago, they were able to diagnose certain types of cancer by specific chromosomal translocations. So there should be a little karyotype image here coming up where they're specifically focused on this, 14 to 18. Specific chromosomal translocation. And this was a long time ago, and they still use it today to characterize these certain types of cancers. That person has a chromosomal translocation. Only after scientists learned how to sequence the DNA and read the DNA letters did they find out what genes were disrupted in this chromosomal translocation. And the answer is going to be that we are going to disrupt the BCL2 gene. Is one of the stories. Okay? All right. So it's an anti-apoptotic inhibitor, and if we disrupt it, what could happen? Think about that, okay? So then I think we got everything went up there. Yep, okay? Yeah, and then, then they just go like this, like BCL3, BCL5, BCL6, BCL7A, BCL9, BCL10. It has clinical significance in lymphoma. So there are other examples of some of these other family members getting disrupted in this type of cancer. They play a big role. How is that coming up? Why is it coming up in this cancer? Remember, those cells are supposed to die pretty quickly. A couple of months if they don't get activated, right? So what about that, okay? So the story of BCL2 began with discovery of the gene's involvement in chromosomal translocations found in, here's our type of cancer, non-Hodgkin lymphomas. And then this thing is very interesting, and it's a new concept for us. Most low-grade non-Hodgkin lymphomas contain this chromosomal translocation that places BCL2 gene on chromosome 18, connected, juxtaposed with the immunoglobulin heavy chain gene. And what's going to happen in that chromosomal translocation is that the BCL2 transcription unit, the normal wild type transcription unit of BCL2, is going to get accidentally connected to the cis-regulatory region of the antibody genes. And remember, it's in B cells, and we've seen before with the MIG story, antibodies, they get expressed at really high levels. This is really strong cis-regulatory region. So we're going to greatly overexpress normal BCL2. What will happen? The cells just won't die. They're supposed to die in just 3 months if they're not activated, and we're making many new cells every day. That's not stopping. So the low grade, I'll just check on these concert tickets. No, just kidding. Is this a thing? Oh, it is a concert ticket, but I won't answer that. I'm looking for the phrase. So what's happening here? Too much BCL2. The cells just don't die. When the doctors look at this patient's blood, they don't have cancer, but my gosh, you have way too many B cells. Why? What do you know? What are you doing with all those B cells? And that's called low grade, and it means it's like the precursor to real cancer. And the person could be walking around like that way for years, where they just like way too many B cells in your blood, but you're doing okay, because you're making too many and you're not killing them fast enough and they're not activated. That's not good. I don't know, maybe get tired or something, I don't know, but you don't really have full-on cancer. So we're talking low grade. And see, that should make sense to us, right? The setup is there. What do you think is going to happen? You're going to get a mutation in something like MIC. It's just going to ramp up the cell cycle and make even more, and they're just not going to be killing the cells. Yep.
So if B cells normally lower an infection, they're not sick until the mutation. Like just having too much B cells, like that's okay. Like it's weird.
Probably have
normal.
a decent immune system.
Yeah.
Going on like that. You
You
just are making too many of them. I don't know how many symptoms a person have or what they'd be like, but I'll just say maybe they're kind of tired, or because their blood is kind of messed up in its composition. Kind of pushing the other blood cells out of the way or something like that, right? So that's what happens. That's what happens first in this is driving high levels of BCL2 gene expression in B cells. These non-Hodgkin lymphomas are turned low grade because the B cells accumulate without having a high proliferation rate, which was an early clue that BCL2 operated in a manner distinct from the oncogenes, right? They're not making extra cells, they're failing to have the unactivated cells die. And just, have to rewind in history back to the 1970s and 80s. The scientists were completely perplexed by this. because the scientists had been discovering those oncogenic retrovirus oncogenes that always stimulate the cell cycle, stimulate proliferation, right? Yep. So, and the BCL2 operated in a manner distinct from the previously discovered oncogenes, such as MYC, RAS, etc., by prolonging cell lifespan rather than driving cell division. It's really obvious, it's really sensible as soon as you kind of jump on what's going on, right? These are B cells, they're supposed to die, they're just not dying. So that's a problem, but it's not a wicked, evil problem. The scientists reviewed their journey and they discovered BCL2. They also explained how they uncovered the mystery of why the B cells express very high levels of BCL2 mRNA protein without genetic changes in the B cell gene structure or sequence, right? It's still the normal wild type transcription unit and protein. And that's surprising too, you know, because you RAS and people are like, look at the mutations in RAS, they're all in the coding region, you know? And BCL2 doesn't have those, you know? It's like MYC, doesn't have those. It's just doing its normal job, right? So for chronic lymphoma, they homed in on another deletion, found the majority of CLLs, leading them to discover the first genetic loss of microRNAs. So that's some extra different thing. So don't worry about that. So this is what we're focused on. And then let's just read a little bit more. BCL2 is localized to the outer membrane of mitochondria. It plays an important role. So that's where we're starting to bring up the idea that it's going to play a role in the mitochondria. We'll see our book talks about that. Important role in promoting cell survival and inhibiting the action of the pro-apoptotic proteins. Pro-apoptotic proteins in the BCL2 family, including BAX and BAC, normally act in the mitochondrial membrane to release cytochrome C. That's what we'll key in on. That's in our book. Important signals in that apoptotic pathway for death. And so they're additional blah, blah, blah. And then let's jump down to damage to the BCL2 gene has now been identified to cause a number of cancers, including melanoma, breast, prostate, chronic lymphocytic leukemia, lung cancer, possibly schizophrenia, autoimmunity. So not just cancer, but roles in lots of different health and disease states. Cancer can be seen as a disturbance of the homeostatic balance between cell growth or cell proliferation and cell death. And so that's why this chapter is here. In this section is we have the cell cycle chapter and stimulation of the cell cycle, followed by the programmed cell death chapter, because there's a balance like that. And that's what we disrupted with this translocation of BCL2. So cancer is being as a disturbance in the homeostatic balance between cell growth and cell death, overexpression of anti-apoptotic genes, and underexpression of pro-apoptotic genes can result in the lack of cell death that is characteristic of cancer. An example can be seen in lymphomas. Overexpression of the anti-apoptotic BCL2 protein in lymphocytes alone does not cause cancer, but simultaneous overexpression of BCL2 and the proto-oncogene MYC may produce aggressive B-cell malignancies, including lymphoma. So, one example, right? That example, non-Hodgkin's lymphoma, and we start out with, you've just got this low-grade one. You're just not killing the B cells as they should, be turned over. Then you couple that with a mutation that actually increases the proliferative rate, because remember that's happening every day, and it's happening pretty fast. And if you get that going with a mutation in something like overexpression of MYC or amplification of MYC, right? Gene amplification of MYC, then you've got a real problem, because you're making too many cells and you're not killing them. That was found here. That's a great example for us to remember this. And then you see this list of other cancers, because in other cancers, you get the cells growing, they should go through programmed cell death. They normally do. And you get a disruption in the programmed cell death pathway, and that just exasperates, just causes more trouble, and the cancer advances. Okay? So, great. And that's really our key thing from the programmed cell death, is to hone in on that. We have the caspases, the effectors, they're doing these actions, these cellular actions, like activating the scramblase and activating the DNases and so forth. And we still have to talk about mitochondria just briefly. And so this is a figure And we don't need to worry about this, but I'll just point this out, that when our book has been showing us these growth pathways, sometimes it's showing us that when growth is stimulated by a growth factor and it stimulates the cell cycle, you're almost always stimulating a change in the balance of the BCL2 gene family members. So the cell kind of starts to lean toward being more pro-apoptotic, or it gets inhibited like that. And growth factors almost always do that. In addition to what we've learned about so much, in addition to stimulating the cell cycle, the growth factor is simultaneously also adjusting the likelihood of apoptosis. And it gets complicated because in some cases the growth factors inhibit apoptosis, and so it's kind of a dangerous game where like stimulate the cell cycle and you're doing great. Keep up the good work. In other cases, the same growth factor layer stimulates the cell cycle and stimulates pro-apoptotic members, and saying you should be worried. You know, this is a dangerous thing that you're up to, and we're worried about what you're doing. And it just is a case-by-case basis, and you know, sometimes it does that, sometimes the other thing, but almost always there's a complex pathway in this signal transduction pathway where it's activating different factors in addition to what we've learned about. So we just need to be aware of that in a general sense. You're not going to be expected to draw out the details of any one of those pathways. Okay? Okay? And here's just an image of this role of the mitochondria. And so you all know the mitochondria is a membrane-bound organelle, and it has a lot of stuff inside of it. And the key thing is just this weird thing, and that is when cells start to go through apoptosis, the membrane of the mitochondria becomes leaky, and stuff leaks out. And one of the things that we'll learn that leaks out is cytochrome C. And cytochrome C leaks out into the cytoplasm. It's not supposed to be in the cytoplasm, and that is part of the pathway to activate apoptosis. And there the situation is, oh man, you've got a problem with your mitochondria. And mitochondria are critically important. There's something really wrong in our cell. So we should probably go through programmed cell death. So you can just see, vaguely here that we have some cell death signals, either internal or external. Then we have these members of the BCL2 gene family upstream of the mitochondria, and BCL2 is in the mitochondrial membrane, and also other places in the cell. And but we get a leaky membrane, and it releases cytochrome C, and that's going to activate some of the caspases, leading to programmed cell death. So that's all we want to get out of that. It's complicated because it includes the pro-apoptotic and the anti-apoptotic members of the BCL2 gene family interacting with components of the mitochondria. But there's a bottom line, and it's really about the mitochondria becoming leaky, the membrane becoming leaky, and stuff leaks out. And the stuff leaks out like cytochrome C. Cytochrome C is just a metabolic factor. You might remember it from biochemistry or from introbio. Just a normal metabolic factor. And so it's not a special thing, but it's wrong for it to be in the cytoplasm. So it's been used as evolved to be a signal to say, we got a problem with the mitochondria. Let's get out of here. Yep. Yes.
So so you said once the cell starts to undergo apoptosis, that's when this becomes leaky. But then it sounded like you said that the leaking of the cytochrome C is what causes the caspase to activate, which causes apoptosis. So what causes which?
Both. So there are caspase activation downstream of the leaky mitochondria. So
Okay.
say, for example, we went back to this figure here. See how this pathway seems really simple, but it includes things like the mitochondria in the pathway, and the mitochondria becoming leaky.
So
So
the
it's-
caspase has kind of started, the mitochondria ramp it up more, so now there's more of it, so now it
Yes.
just does it quicker.
That's a good point too. In this is that question Sam asked about when we get the red light to turn green,
Oh,
how does it turn fully green? Because it starts to flicker. And I use that example in P27, seeing, well, there's a feedback loop that's a positive feedback loop where it starts to flicker green and then it goes full green. And you know, the cars are like, yeah, that's a green light. Same thing here, there's a balance. And so like when I say we're leaning towards pro-apoptosis, we're leaning toward anti-apoptosis, it's a balance, kind of like a light flickering. I don't want to get these muddled, you know, but kind of like a mixed signal. And then there are these positive and negative feedback loops that are just going to either accentuate it and make it stronger or weaker. And so that's why the book is saying this here, because it's showing that the cat spaces are also involved in mitochondria. How's that? I just said they're mitochondria is upstream of the cat spaces. And the answer is, when the cat spaces get activated, you're dying. And so how do they kill the cell? Flip the lipid, the phosphatidylserine out, you're going to get eaten. Just chew up the DNA, and also just enhance the destruction of the mitochondria. Just be like, just dump everything out of the mitochondria. You're out of here. Yeah. So it's a feedback loop like that, right? So it's the cat species are both downstream and somewhat upstream of just making the mitochondria get all messed up. Yep.
Once the pro starts, can any of the anti undo it, or is it the triggers?
Once the caspase is rolling, it's toast.
Okay.
Yeah, yeah. The caspases are the effector proteins. You know, the gun has been shot, and you're like, oh, I'd like to take that back. Yep. Yes. So that's that thing, and I think we're really close to being telling the package that we really want to tell. Let me just see. So we have that one very good example of the non-Hodgkin's lymphoma to fit in, and then, yeah. Yep, so that's really what we're going to talk about the chapter. So, the chapter comes into two parts. One is the stem cells, and we know something about stem cells, and we're adding to it. So that video is really important because it's, got stuff we already know, and we're building on it, right? And then this program cell death, and it's a different story. But in our example, and in lots of examples, it ties together with cell cycle regulation. So sensibly, logically, like that. So let's just pause, see if people have questions. Yep.
I have a question on the stem cell video, the last one, the induced pluripotent stem cell.
Yep.
My question is, when you're making the transgenic genes that you're then going to put in our example of the muscle gene, you are not taking the muscles, because it's like, I know that they would be, I know we talked about how we had to like undo methylation and everything. Are you taking the muscle cells, like let's say MYC gene for instance, unmethylating it, making it a transgene and putting it back in? Or are we taking another cell's genes and then putting them into the muscle genes after?
Yeah. Yeah. So in the stem cell video assignment, I talked, well, there are two normal type of stem cells in our body, adult stem cells like the muscle differential cells, and then some embryonic stem cells that I don't think we've talked about in our course. Then there's this third example, and it's an example where scientists can make stem cells, can turn cells into stem cells, and they're called induced pluripotent stem cells. So that's an experiment. That's research, and that's an experiment that doesn't occur in nature ever. And the logic is just this. Oh, we've been talking about that cells have genomic equivalents. They have all the genes. So you should be able to, in theory, just take any cell and turn it into a induced pluripotent stem cell that could make a whole nother organism. It's got all the genes. And just pick one, pick a differentiated cell. And a key concept is, well, what are we going to have to do to get that differentiated cell to be a stem cell? And the answer is, it's got all the genes, it's methylated. It's done DNA methylation on a lot of those genes. So one thing we're going to have to do is we're going to have to remove a lot of that DNA methylation so that all the genes can be activated again. And then another thing is differentiated cells have almost always left the cell cycle, and they're in G0. And this is where we remember that G0 is not a big detour. In reality, those cells are sitting at the red light. But we'll need to turn it green again because our brain cells, our muscle cells, they're not dividing again in our lifetime. We got to get them dividing because a stem cell has to be able to divide and has to be able to express all the genes. So scientists, as described in the video, say, Well, let's figure out what genes are expressed in stem cells, because there must be some stem cell genes that are expressed, and instead of trying to mess with the existing cells, we will just take some cells and we'll manipulate in a test tube and we'll put them in. So we'll add different cis-regulatory regions so we can force the expression of those genes, right? Those stem cell regulatory genes in a muscle, they've probably been methylated. So, you know, you can't escape the thing, right? You gotta demethylate them, but how are you gonna do that, right? So the plan is to do this, and it's confusing to students. It's take some normal genes from a different cell source and build transgenes in a dish, put those into the differentiated cell, and these new transgenes will force expression of these proteins that would normally be found in the genes, but it's just too hard to activate those because they've been methylated, shut down, something like that, right? And so that's what's confusing, and you're asking about that, is you take another cell source, get the DNA from that, and manipulate those genes. There's just really a focus on 4 genes, and one of them is MYC. And so they're going to take a MYC gene out, just put a different regulatory region on it, and put that in any differentiated cell, and just pushing MYC that hard, make the cell go back into the cell cycle. Is that cell off in some detour, G0 detour? No, it's just sitting at a red light. What's it need? Cycling. MYC can make cycling, push it, you know, like that. It's really amazing. You know that that's a dangerous game, right? They're doing it in a research lab, but are they going to put that back in the human body? That'd be pretty stupid because you're just pushing MYC to drive the cell cycle, you know? And you're like, you know, what? It's going to cause cancer. That is cancer. You're putting in cancer cells. So there's a lot of research that's going on, right? The discoveries were made now 15 years ago, and there's a lot of research going on to try to learn how to activate the cell genes differently, right? Or turn the regulatory or turn those genes on that, you know, we're pushing the cell back in the cell cycle, but then have some control over them like that. But yeah. And so and that's in that video, and it should make sense, right? The scientist's last name Yamanaka, and working in Japan, you know, basically 15 years ago, like that. And his labs figured this out on their own. And the key thing is, when you get to this point in this course, you could have figured that out. You'd be like, hey, Professor Champlin, like, if a cell has all the genes, why couldn't it be a stem cell and make a whole new organism? Or we don't need to make a whole new organism. Why can't it just be done where you could make a new kidney that would be that person's kidney? You know, they wouldn't get a transplant. They would just get a new kidney of their own growing in a dish. Plop that in, you know? So you could have thought of that, right? And, you know, like when you get up to speed on these topics, the genius ideas are not, you know, like Albert Einstein ideas. They're like, well, we should figure that out. How are we going to do it? And the answer is, well, let's look at stem cells. Remember, there's the stem cell. It's going to divide in 2, and it makes a self-renewing stem cell. That's a stem cell. And then a precursor cell. That stopped being a stem cell. Both cells have all the genes. What genes are expressed differently? What mRNAs can we detect in stem cells, but not in precursor cells? Because that must be how a stem cell stays a stem cell and a precursor cell stops being a stem cell, is the precursor cell stops expressing these stem cell genes. Find those genes and then manipulate the DNA for those in a test tube and just force the cell to express those genes. See what happens, yeah? Yeah. So it's, you know, it's hopefully that's kind of satisfying. We're like, oh man, if I was born 15 years earlier, I would have a Nobel Prize, you know? You can think that, right? Yeah, it's just like that, right? That Bob Horvitz that did all the work at MIT in Boston, I did a job interview to work in his lab. And when I was doing the job interview, I told him I was just going to lie down on his couch because I was pretty tired. So he's talking to me, this guy that's going to win a Nobel Prize, he's talking to me. Am I a loser? Yeah, you know what? I'm a loser. Because I was just like, you know, it's a job interview, but I think I'll just rest on my back on your couch. And he was surprised, you know, really surprised, but I didn't care. Yeah, driving in Boston the first time in your life. If you grow up in the Midwest and you have to drive in Boston your first time, and the entire exhaust system is falling off your Ford Escort, but won't fall completely off, is really tiring. And you just want to get to the job interview and just lie down. So anyway, let's do a little concept chat about what we thought were important concepts today, and we'll do that quickly and try to meet another person.
I grew up in Nebraska, and when I drove to Boston the first time, I paid like $100 for someone to take my car and park it, and then
Yeah.
I bought a bus pass.
Yeah. No, Boston is really like,
You get into the tunnels and you're like, oh my god.
just on any street in Boston, you're just like, and the Boston drivers are like, I can take a left turn in front of
And
you.
your maps start working when you go into the tunnels. Okay.
One thing I thought was pretty cool was scrambling. Yeah, how the effector.
What is it? Cutting the. I understood it was cutting something because after I cut it, then it became active. But what is
Like a
this?
protein type of rope in that shape. You can just cut off a chunk that makes it.
But what protein is that? I missed. Like I was lost.
Scramblers.
That's scramblase?
Yeah.
But so what scramblase is what's gonna convert. Oh, okay. That's the protein that sits in it, switches the phospholipid.
Yeah.
Okay, makes sense. The other thing I thought was really interesting was the BLC2. That is an inhibitor of caspase. But it's like, yes. But it's from a whole gene family, and basically you have a bunch of them that are either going to inhibit it or inhibitive, and once caspase is, correct me if I'm wrong, this is my opinion, once caspase is getting started, it is going to cause the cell to start to undergo apoptosis in the BL and the BCL2, it might be a less training network, but then that's going to cause the mitochondria to start leaking, mitochondria is going to leak the whatever C stuff.
BCL2 is an anti-apoptotic.
It's an anti, so it's against that.
So it's making it so it doesn't make apoptosis.
Okay, but we're talking about a pro one, I guess. So we're talking about a pro one.
Which is the family in general, right?
Yeah.
The family has activators like
Right. I do have an inhibitor, but this inhibit, the inhibit function has to happen before, like this is upstream of the cascade before you start This is like telling yourself, like, calm down, you're okay, don't kill yourself, don't jump off the ledge.
Right, and when it's mutated, it overproduces into something that works.
My understanding was after the B cell met its target, that's how it never died. Once it was no longer yeah, but like I thought that was like once it was activated. I didn't think that had anything to do with caffeine.
I'm just talking about like BCL2 in general. If it isn't like mutated like trans.
We'll just do this for another minute.
It will.
Oh yeah. Like you had too much of it. You know what? I love that. Okay. It's not a problem.
That's gonna be
No, I can understand growth factors. Can you explain to me? Because there's like sometimes growth factors inhibit.
Oh, yeah. Okay, let's check in with Teresa. Teresa, what is one thing your group talked about that you thought was important today? Oh, isn't that Teresa? No, is that Lilith? No, there's Lilith. Oh, Mariah. Sorry, Mariah. Yeah, got you. Mariah, what's one thing your group thought was important?
We talked about CDK and caspase both being effectors.
Yeah, and you know I've emphasized that effector term is it's a kind of a good term to appreciate the difference between the action and the upstream regulation, but it's a kind of a context-based thing. So like we saw that with the cell cycle, where sometime we would say, what's the rate limiting step of the cell cycle? And we might say, well, it's the lack of cycling. But then we would zoom out, because under the skin, the real rate limiting step is actually the PDGF, which is going to increase the cycling. So it's not really the cyclin that's a regulating step in that. So that effector story can become like that too, where the attention can shift to the place where it's really the effector, that's really the the action item kind of thing. So it's a little bit frustrating term, but as long as you recognize that, it's a nice term to appreciate. We've reached the end of the road. When we activate caspase, this cell is going to die. And it's satisfying from, you see that cell biology of it, because you're like, yeah, well, it's just going to flip those phosphatidylserines out. So, poosh, that cell's going to be eaten very, very soon. And then it's chewing up the DNA, it's making the mitochondria leaky. It's just really, the actions are death actions, right? The nucleus is just falling apart, And that's kind of satisfying to appreciate. We got all this regulation, but here comes the action. It's also nice to think about that and appreciate that, there are so many of these regulatory proteins. That's all they do. This is MYC is all MYC is doing is transcription factor turning on some other genes, and yet it's, an oncogene. It's a very deadly gene, but it doesn't really do that kind of action that you'd be like, whoa, that's big action, It's kind of interesting to appreciate that regulatory proteins and factors can have that bigger role, like that. Sam, what is one thing your group talked about?
We talked about the idea, we talked about a couple things. We talked about the P53 linkage through the BCL2 family to grow.
Yeah, man. Like we shouldn't get tattoos. We should open a tattoo parlor and be like science tattoos, biology tattoos. Come on in, right?
Sure.
Yeah, I bet if we took over that Suntiki just off campus, have a tattoo parlor, we could get students down there after class. Just come on down after class and we'll help you with, and then you're just cheating on your exam by looking down at your arm. You're like, I had to take this exam. And they're like, you have to wear a long sleeve shirt, and roll them up, right? Like that. And then let's see, I also wrote down Matt, what is one thing your group talked about?
We were discussing how caspases cuts scramblase.
Yep.
And just takes off that inhibitory piece.
Yeah, so again, proteases are enzymes that cut proteins, and there are lots of different proteases. Some of them just fit into our metabolism for the proteins that we eat in our diet. We're going to break those down into individual amino acids. And so those proteases are general proteases. Lots of proteases have very specific sites where they cut, between specific amino acids and during, lengths of amino acid sequence, so they'll cut right there. That's a big important way how the complement cascade gets activated when our blood starts to clot. Those clotting factors, a lot of those are precursors that need to be clipped like that, proteolytically clipped. And so caspases are proteases that clip target proteins and thereby activate them. Yep. Questions?
This might be a really dumb question, but I thought that proteases removed a phosphate group and endonucleases are what caused the random cutting.
Okay, so that's mixing some terms. And so proteases are always targeting proteins for cutting.
Okay.
The thing that removes a phosphate is called a phosphatase.
Okay.
Yep. And then an endonuclease
Cuts
is a DNA enzyme that cuts DNA, and it cuts it in the starts to cutting it in the middle, like scissors cutting in the middle or along the length of it, rather than an exonuclease that chews in from the end.
Okay, back to my flashcards again.
Yeah, and that's really legit, right? To just self-quiz as you walk out of class, and or driving around, is to just think, well, what can I remember about BCL2, and that's just good because you're recalling, and that's how you strengthen you're practicing singing a song and being like, you like that song, I want to sing it, like that. Okay, so we're all set, and Thursday we'll roll into the chapter 19 that's going to be on our exam, and it's a lot of stuff that's got a little bit of familiarity to it, so we'll be able to deal with that. And we'll be in good shape for the exam. It should be kind of familiar territory, heading toward a familiar exam format. All right.